Dear viewers, welcome to this session on green biotechnologies in the field of waste management and bioremediation. I introduce myself as Dr. Sushmita Bhaskar, Assistant Professor in Environmental Studies from the School of Interdisciplinary and Transdisciplinary Studies, Indira Gandhi National Open University, New Delhi. So, after listening to this lecture, you must be able to understand the various sectors where environmental waste and waste pollutants are being released and how these could be tackled with green solutions, and we can also give an account of the various microorganisms that are used for bioremediation purposes. Our environment, it is a natural habitat for a number of organisms. We have the tropical forest, we have the coral reefs, then we have a very long coastline. For example, India alone, we have a coastline as long as 7,500 kilometers, and this is home, so there are so many wetlands which are also identified as the Ramsar sites and the heritage sites, and we have a number of organisms, unique and endemic organisms, endangered, and a number of measures are being taken in order to protect these organisms. Again, we have the coral reefs. So, all these are being threatened from the pollutants from human activities. We have natural resources, coffee, tea, the spice garden of India in Kerala. So, we have all these natural resources that are produced, and our environment, again the different ecosystems, they are able to hold. For example, if we talk about just one of the systems like the wetland or the water systems, they are kidneys of the landscape and they are cradles of biodiversity. So, in the Western Ghats, if you see the newspapers, every day you find new species of frogs or other tropical species that are being discovered by scientists. So, all these are advantages, and again we have the ecosystem service functions that our environment provides us with. They are for the provisioning services, regulatory, and they also help us from the climate change processes, and for the biogeochemical cycling of the elements, and they also provide sustainable livelihoods for millions of people. So, for example, if we again take one of the sectors like wetland, many of the communities living in the Andaman, they can grow these crabs which grow over there, and when they import it or export them to the other countries within India, of course the amount that they get for a kilo would be less, but when they export them to other countries, maybe nearby in Thailand and so on, then even for 1 kg they can earn a livelihood of up to 20,000 rupees. So, all these are certain sustainable livelihood opportunities also that our environment is giving us. But in the recent years, past decades, we are seeing that the environment is heavily being polluted just because of man's selfish needs, from the various industrial activities, technological applications. We need to put an end to all this, and we need to go on to the greener technologies. So, in this figure, that is what it aims to, it is self-explanatory, and that is what we aim to explain. The garbage, the trash, the plastic trash also, even though the plastic has been banned, but still we are using the plastics. So, all this is choking our planet. Industrial waste and industrial pollutants, you see the plumes of the air pollutants that are being released into the air, the spraying of hazardous toxic chemicals onto plants. So, all this is choking our planet, and also irreversibly human beings are affected with various health ailments. If you see, it is the diabetic nation. All this have the problems related to the environment and health. So, the various environmental conferences like that which took place in Stockholm in 1972, the Rio de Janeiro conference, and also the United Nations World Summit conference that took place in Johannesburg in 2002. All these have been time and again trying to depict the picture of the real environment and how we are living in. And it is high time that human beings take action for sustainable development goals, and these are being defined, and we are also progressing and marching on to these lines. Certain environmental concerns which are there is, we live in a highly industrialized world. There is too much of technological development, there is depletion of natural resources, there is depletion of the global ecological capital, and we are marching actually from unsustainable to sustainable development. That's how we need to go, but actually it is a reverse trend. We are actually going on to unsustainable form of development. And if we don't go in the right direction, then that will lead to the doomsday that was written in the Limits to Growth by Meadows et al. In 1972. So, for this the local public, the government, the NGOs, institutions, everyone, all of us need to come together in order to tackle the problems of environmental concerns. Some examples, I will give you two examples in two different industry. The first one is a tanning industry. So, here chromium is used as an agent, as a basic chrome sulphate. It is used in the tanning industry, and for the leather and processing of the hides. It is toxic and it is a known and potent carcinogen which causes cancers. The hexavalent, so this comes in two forms. You have the trivalent form as well as the hexavalent form. The hexavalent form, that is chromium 6, this is most toxic and most soluble even in the water. It induces free radical formation and it induces oxidative stress, DNA and membrane damage. In Kanpur, if you go that entire belt of Moradabad and the Kanpur belt, you find the predominance of the leather industries, and 380 odd leather units are located in the Jajmao area right on the bank of the river Ganga. And this is the single largest contributor to pollution of surface as well as groundwater in this Jajmao area in Kanpur. Now, these two forms of chromium that we explained. So, chromium, it is very easily, it exchanges the state also. So, it can change from the tri to the hexa according to the environment around them, according to the pH condition, the alkalinity and also the acidic conditions of the environment, microenvironment surrounding them. So, the hexavalent form is extremely harmful to health, and in Kanpur they use the chromium sulphate. Still the chrome recovery plants in some places they have been installed and some not, despite the court's orders. This example, in the photo you can see that the groundwater is totally yellow in color, and that is a chromium color that has come in the groundwater. So, in Norakhayda, it has been used as a waste disposal site by the basic chrome sulphate manufacturing unit, and they supply this BCS to the leather industry. And the people are drinking the water. You can see from the well, from the pump, the groundwater is coming as a yellow color with chromium, which is a carcinogen. So, it is understandable that the water is laced with toxins, and this waste water, they are using this for irrigating the land also for agriculture. 2,500 hectares of farmland, this water is being used to for the irrigation, and this water is highly toxic because it has heavy metals such as chromium, arsenic, lead, mercury as well as cadmium. You can see the people that they are affected with various skin problems, and all this is due to this toxic chromium which is affecting their health. So, that was one industry. Now, coming on to the textile industry. So, India, if you see, we all love wearing colored clothes, and where do all the colors come from, or these beautiful hues? They are all from the dyes. So, azo dyes, these are the dyes that are used in order to color our clothes and to make them attractive and nice. So, the textile industry is a major sector in our country, and most of the pollutants are they are released. The textile processing used a number of chemicals that can be toxic and harmful to human health. In India alone, we have more than 2,000 textile industries, 83 composite mills and 2,241 semi-composite and processing units. Maharashtra or Bombay is also known as the Manchester of our country, and Coimbatore is known as the Manchester of South India. So, Tamil Nadu, Maharashtra, Gujarat and Punjab, they are the 81% of the total textile industries in the world, and these fibers that are obtained from the industries, then they are made into yarn. This yarn is then converted into fabrics, and then various dyes, chemical agents, sizing agents, finishing agents, also bleaching agents, all this are used in the different stages of the production in order to produce the final product of the cloth. So, this waste, in some cases the industries do treat the waste before liberating them into the nearby streams or the environment, but in certain cases even the cottage industries, they do not treat the waste, they are just as such they are entering the different environments. So, that waste release can be hazardous to the environment as well as to the human health. Azo dyes are used, which are synthetic organic compounds, and they are widely used in such textile units. These effluents contain high concentration of the dye, and the breakdown products are persistent, which cause toxicity. So, again this is also not degradable and it remains in the environment for number of years. So, now that we have seen two case studies, one of the tanning industry and the other of the textile industry, do we have some green solutions, green biotechnological solutions for the bioremediation of such waste? Microorganisms and plants, they have the ability to detoxify this waste, and the mechanisms include both genetically engineering microbes and isolating the naturally occurring xenobiotic degrading microbe. So, there may be a place which has the contaminant, and from there itself you can isolate a microbe and then use that in situ as well as in vitro techniques, and then use them for pollutant removal. The heavy metals can be removed, for example, the mercury, cadmium, chromium and lead. So, in the scientific experiments that have been conducted in the laboratory, they have been found to be very useful. The different techniques that could be used include the microbial remediation, the phytoremediation, photoremediation and other techniques which also involve newer and novel technologies. The degradative organisms, they can be engineered to increase the mobility in order to access these compounds, including enhanced chemotaxis. And one of the limitations of the bioremediation process is that optical conditions are required, because any microorganisms, it requires the proper temperature conditions in order to grow and survive for its metabolic processes to operate and to be intact. So, this is one limitation that the bioremediation process faces. Now, we could have ex situ, in situ, the above ground strategies where bioreactors, biosorption, biopiles, bioleaching, phytoremediation techniques could be employed. We could have bioimmobilization, soil washing, infiltration galleries, bioventing techniques that could also be used. Now, giving an example of in situ bioremediation process, here we can have bioattenuation, biostimulation and bioaugmentation. So, here we can monitor the natural progress of degradation so that the contaminant decreases with a time. The biostimulation, where the stimulation or intentionally we are stimulating the resident xenobiotic degrading bacteria by electron acceptors, water, nutrient addition or electron donors. Bioaugmentation is the addition of lab grown cultures which have been proven useful in degrading the xenobiotics, which have their biodegrading ability. So, this also can be used for the bioremediation processes. Now, we come on to the certain microbes that are useful in the bioremediation process. Aerobic bacteria like Rhodococcus, Gordonia, Bacillus, Moraxella, Micrococcus. We have anaerobic bacteria like Syntrophus, D. sulfovibrio, Methanospirillum, Methanoceta. All these have been useful for degrading xenobiotics. Methanogenic and methanotrophic bacteria, so that is they produce the methane or the methanogens, they are known as the methanogens. Methane monooxidase, oxygenase enzyme that is generated by the methanotrophs. This can degrade a wide variety of the organochlorine and the chlorinated hydrocarbons. So, again these are very useful in bioremediation. Certain blue-green algae like the cyanobacteria, they can degrade oil derivatives. Sphingomonads can degrade wide range of xenobiotics, synthetic polymers, aromatic compounds, and due to its plasmid bone mechanism, they are found to be very useful. The new technologies include identification of the genes responsible for the specific genetic like xenobiotic compound detoxification or degradation, and that can help in the forming or the developing the recombinant genetically modified organisms for the remediation of the complex waste. Some more species like Pseudomonas, they are useful in the degradation of organophosphorus pesticides and fungicides, methyl parathion, aromatic and aliphatic hydrocarbons and oil degradation. Pseudomonas species, again they are used in the remediation of heavy metals and phenolics, for reclamation of oil and metal contaminated soil by producing surfactants. Then Bacillus species are able to degrade benzamidazole compounds. Azotobacter can remove the chromium through biosorption. Then textile effluents and also the polyethylene. So, they can be acted upon by the Anoxybacillus rupiensis and Brevibacillus species, also the Rhodococcus species. So, all this can be used for degrading even the plastic waste. Microbial enhancing oil recovery processes, they use the biosurfactants that reduces the interfacial tension between oil and water interfaces. So, Bacillus subtilis, this releases surfactant. It is a biosurfactant used in oil degradation. We can also employ the use of fungi, that is in mycoremediation. So, this is using fungi for bioremediation of contaminated environment. It can degrade petroleum hydrocarbons, polychlorinated biphenyls, heavy metals like biosorption, the phenolic derivatives, persistent pesticides, ligninolytic fungal degradation, fungal biosorption, mycorrhizal fungal degradation. So, these are some of the types of the mycoremediation that can be practiced. Ligninolytic fungal degradation. So, this can be accomplished by the Basidiomycetes, Ascomycetes. They can degrade lignocellulose material that is present in the dead wood and the paper and the pulp effluents. So, this is very effective in the paper and the pulp effluent industrial waste. Fungi can also work on the mechanism of biosorption. So, they passively concentrate and bind the contaminants onto its cellular surfaces. So, the mucus species, Aspergillus species, Rhizophus, the Saccharomyces cerevisiae and also phanerochaete species, they are useful in the heavy metal biosorption processes, and the Saccharomyces biosorbs heavy metals such as lead, gold, cobalt and also copper. Mycorrhizal fungal degradation. So, mycorrhiza, this is a symbiotic association between the fungi and actinomycetes with the root zones of the vascular plant that can increase the soil organic carbon. So, certain species that are of these symbionts or the mycorrhiza are the Morchella conica and Tylospicno fibrilnissa. So, this is useful for the mycorrhizal fungal degradation processes. And the next mechanism is the phycoremediation. So, phycoremediation can be accomplished by using the algae, microalgae and macroalgae. They can remove and biodegrade pollutants from the environment. There are certain special polysaccharides in the cellular walls, in the cell walls that have the metal ion binding sites. Certain species that are found to be effective include the Chlorococcum, the Chromococcus, Desmococcus, Dactylococcus and also the Chlamydomonas. So, these species are beneficial. Also, we find the blue-green algae used for the wastewater treatment, spirulina used as a biosensor to detect the mercury concentration accumulated in the solid waste residues and also in the soil. Phytoremediation, this is also known as green remediation, botanoremediation, agroremediation and also vegetative remediation. So, this uses green or higher terrestrial plants for treating polluted and contaminated soils. Certain plants that have the deep roots and luxurious leaves associated bacteria in the root zones, they allow the plants to absorb, take up and also accumulate or sequester these heavy metals and they can also degrade the contaminants in the environment. So, this picture, that is what it shows, that even plant roots can sequester the heavy metals such as cadmium or nickel and zinc, proving solutions for contaminated environments. The phytoremediation can be classified into certain categories. This includes the rhizodegradation, enhancement of naturally occurring biodegradation in the soil through plant roots, phytodegradation, uptake and metabolizing and degradation of contaminants within the plant by enzymes produced and released by the plant. Phytoextraction, so this is also known as phytosequestration. So, in this there is uptake of contaminant by the roots and then subsequent accumulation in the above ground portion of a plant. So, it is also known as phytomining or biomining. Certain species which are found to be effective include the Brassica juncea, Burkea, Alyssum species, Thalapsi and also Thalapsi geosingensis species. So, they are all useful in the phytoextraction processes. Finally, we also will deal a little bit about rhizofiltration. Removal of contaminants by plant roots in the surface water, in wastewater or extracted groundwater. This could be done by this technique. The process includes adsorption, absorption and also precipitation onto the roots. The sunflowers are useful in this radioactively contaminated sites. So, they use this process of rhizofiltration where sunflowers are found to be very effective. Phytohydraulics, this is also one technique that is used where the use of deep rooted plants can degrade the groundwater contaminants that come into the contact with a root. So, in this case the plants are really deep rooted and that root can bring about the detoxification and degradation of contaminants. Phytostabilization, the use of plants to immobilize soil and water contaminants. The Indian mustard and endophytic bacteria such as Pseudomonas, Burkholderia species, Bacillus and Azospirillum are useful in this process. Summarizing this lecture, so in this session we have understood the importance of the various types of bacteria, the fungi, the mycorrhiza, the symbionts, and phytoremediation, also the novel technologies that could be applied for remediating the environmentally contaminated places and the soils that have been contaminated, wastewater sites by the industrial effluents, industrial waste which are treated, not treated, radioactively contaminated sites. So, we have an overview of the green technologies, green biotechnologies that could be used and the eco-friendly alternatives for treating waste in environmental management. Thank you.